Welcome to Brain Ponderings, your window into the world of brain research and related research. Today, it's really my great pleasure to talk with Richard Johnson. He's a professor of medicine emeritus now, I guess,
Yeah.
at University of Colorado School of Medicine. He was previously at University of Florida for quite a while, I think. And he's an expert on fructose. He's an MD. He's as as our viewers and listeners will know, uh, there's a lot of high fructose corn syrup in drinks and other things, and there's a lot of evidence it's not bad. And Rick will tell us why that's so, what's going on when we ingest fructose and why it matters, and it's very interesting. And then I'll pitch in a little bit about the brain because we did a little bit with fructose. It's bad for the brain too, so I'll talk about. So welcome, Rick.
Yeah, thank you, Mark. It's really great being on your podcast. And yes, high fructose corn syrup is a bad type of food. It's one that we should really limit. And also table sugar. And table sugar also contains fructose. Table sugar is a disaccharide called sucrose, which contains both fructose and glucose. And we have done some work on fructose in the brain, and also I've proposed that fructose has a major role in Alzheimer's disease. And so maybe we can talk about that during this podcast. But yes, I'm a physician. I practiced medicine for decades as well as did research. I've been doing research for more than 40 years. I'm still doing some research. I've got, and so, kind of like you, Mark, I got infected with the research bug, and I've been trying to discover, look at big issues and try to understand why we're getting the diseases we get.
Yeah. And so you're back, are you a Midwestern person?
Yes, I am. I was raised in Madison, Wisconsin, and went to medical school in Minneapolis. But after that, I spent like 20 years in Seattle during my internship and residency, and then I went to Baylor in Houston, and then Florida,
Hmm.
Gainesville, and then Denver. So
Oh,
yes, I've
okay.
had quite a circuit, and originally Midwesterner.
Yeah, I'm from Rochester, Minnesota, so
Oh,
not too
fantastic.
far away from Madison.
Yeah, there you go. Yeah.
Yeah.
And we're probably going to move to Chaska, which is pretty close to
Oh,
Rochester.
oh, really?
Yeah.
Good. Yeah, it's a nice place to live.
Yeah.
And the winters are getting milder
Yes.
with the climate change.
Unfortunately.
No, there's not much snow there anymore.
Yeah.
Um, so then, and you've written, you can talk about your research, but you've also written some books I see, The Sugar Fix, The Fat Switch, and I guess the recent one, Nature Wants Us to Be Fat. And that
Yes.
those are all related to fructose.
Yes. So our work suggests that fructose is involved in a major mechanism to cause obesity and diabetes, as well as neurologic disease. But originally it was aimed in an evolutionary sense to be beneficial. But what happened is what's evolutionarily beneficial doesn't involve taking fructose in the kind of concentrations that we're currently doing. So in our current society, maybe 15% of our diet is sugar and or high fructose corn syrup. And some people are eating as much as 20 or 25% of their diet from these added sugars, and we're just not able to cope with this amount. And what was meant to be beneficial is turning into something that is actually horrific. So, I believe that these high concentrations of fructose are playing a major role in many of the diseases that we're involved that are afflicting us.
So if we say vegetarian diet mostly, some meat, some fruit. and nothing else, right? No ultra processed food or simple sugars. I guess the main source would be fruit, right? Of fructose.
So historically, if you go back to the days before sugar was discovered, sugar was discovered actually from the sugar cane, probably 300 or 400 BC. But if you go before that, really the only fructose that people were exposed to in the diet was from fruit and honey. But when sugar was introduced, it rapidly was loved by whoever tasted it, and pretty soon it became a desired substance, and sugar production went way up. And so today, at one point, yeah, right now we're eating somewhere between 120 and 150 pounds of sugar a year. And when I say sugar, I mean both high fructose corn syrup and table sugar.
And it's
So there's
addictive.
been a huge change. Now, we did have a big discovery a few years ago, which is that um, a lot of the foods in our diet can be converted to fructose in our body. So it turns out that it isn't just
Hmm.
sugar. And what we discovered was that um, the, and I should preface it by saying that it was known for a long time that glucose, which is the other simple sugar besides fructose, remember, glucose and fructose are bound together to make table sugar, and they mix together to make high fructose corn syrup. And glucose is the main sugar in our blood that we use to help. It's the main carbohydrate fuel, and glucose is used a lot by the brain, for example, as a fuel. And fructose is not typically very high in our blood. It's very low concentrations, but glucose can be converted to fructose, and likewise, fructose can be converted to glucose. So under certain circumstances, we can convert foods that you know contain glucose. We can convert those foods to that glucose to fructose in the body, and the main way that happens is through a specific enzymatic reaction. There's only one way it can happen. It goes through in mammals and in humans, and that is called the polyol pathway, in which glucose gets converted to fructose. And it's the primary stimulus for that conversion is when glucose concentrations go up in our blood. So in a normal, like a normal blood glucose concentration, maybe like 80 to 100 milligrams per deciliter. This is, our normal glucose. But when we eat pancakes or bread or rice or potatoes, what we call high glycemic foods, when we eat these foods, the blood glucose goes up, and then it can be converted. That activates the enzyme that converts the glucose to fructose. And we have some evidence that happens. There was even a guy who showed that it happens in the brain. You probably know Sherwin at Yale, who used magnetic resonance, nuclear resonance to measure fructose in the brain of humans, and showed that when your blood glucose is high, you start making a lot of fructose in the brain. So what turns out that when we were studying this science, we came to the conclusion that there's two major mechanisms for fructose exposure. One is through diet, and it's mainly the big exposure is sugar and high fructose corn syrup, and you can get that in a super rapid way by drinking a soft drink. But the other way is the body can make fructose from glucose, and when the body makes it, it isn't just in the liver. It can be in the brain, it can be in the kidney, it can be in the heart, and it can be significant enough to cause disease in any of those organs. So we've been able to show that this, what we call endogenous fructose, endogenous fructose may be even more important than dietary fructose under certain circumstances. And
And Rick, how did you show that?
yeah,
How did you had to block endogenous fructose production somehow, right?
yeah. So the way we showed this was we had to understand how fructose works, and there's a biologic pathways that we can talk about, and then there's the biochemistry pathways. But basically, when fructose is metabolized, there's a specific enzyme called fructokinase. And fructokinase converts fructose to a fructose 1 phosphate. And that chemical reaction triggers a whole series of events. And the fructose 1 phosphate can be continued to be broken down and enter into the classic glycolytic pathways that lead to energy production. But fructose 1 phosphate can also act as a signaling agent and signal different enzymatic act. It can activate, for example, it activates glucokinase. And this enzyme uh, generates its own set of things and can drive other enzymatic pathways like the pentose phosphate pathway and the polyol pathway. Whereas also when the fructose is converted to fructose 1 phosphate, there's this ATP depletion. ATP is the energy in our cells, and it is consumed rapidly, but then it can generate byproducts like uric acid, and the uric acid can act as a signaling molecule itself to stimulate NADPH oxidases and other things that can cause mitochondrial issues. Anyway, we spent a lot of time doing the enzymatic pathways and looked at that. And then we also looked at the biologic pathways, and we could show that, fructose caused mitochondrial oxidative stress, for example, and unregulated glycolysis and all these things. And that led to, fat generation, all that. So we could show the enzymatic pathway that was unique to fructose and distinct from other sugars. And then we could also show the biologic effects in animals by giving fructose and showing, that they develop obesity and so forth, and identify how they're doing it, and then look at other animal species and so forth besides the mouse and do clinical studies. And then we could prove that it was this pathway by knocking out the enzymes in the pathway, whether it's fructokinase or other enzymes like AMP deaminase or any aldolase enzymes involved in the pathway. And we could also selectively knock it out. So we could selectively knock it out of the liver or the intestine or the brain, and so this and I've probably published 300 or 400 papers on this area, maybe more actually. And so we can go into individual ones, but what we discovered was that high glycemic diets like pancakes, like rice, like potatoes generate fructose in the body, and it generates it in the brain, and it generates it in the liver, and it generates it other places. And that fructose is enough to cause disease, because when we knock out fructose metabolism, for example, in an animal eating glucose, we can block the development of insulin resistance or diabetes, or we can block, maybe half of the effects to cause obesity. So we were able to show that it wasn't insulin that was the big problem. The problem was fructose. And
So
so
when you have high fructose in the
powerful.
cell, high fructose in the cell relative to glucose, there's a relative reduction in ATP levels. It's
Oh yes.
yeah.
Oh,
And
absolutely. Yes.
is there a role for mitochondrial uncoupling in that and heat generation? In other words, when if you have high, I think anyone noticed it. If you eat sugar or high carbohydrates, you actually feel hot. So I'm wondering if there's mitochondrial uncoupling occurring.
There's an acute thermogenic effect. Yes,
Yeah.
there's an acute thermogenic effect, but it's just acutely when, as the ATP is being consumed rapidly, and there is some heat generation. What I will tell you is that the main effects of fructose on the mitochondria are through inducing oxidative stress to the
Mm.
mitochondria. It actually leads to a translocation of an enzyme that produces oxidants, NADPH oxidase, into the mitochondria. And there are certain mitochondrial enzymes that are quite active and that are inhibited by oxidative stress. And the most famous is one called aconitase in the Krebs cycle. And by inhibiting that, it leads to citrate accumulation, which stimulates fat production. It moves into the cell. So there are all these different things. It blocks fatty acid oxidation. So it blocks the burning of fats. And it does that by inhibiting several enzymes, including the transfer of fatty acids into the mitochondria, but also by blocking the fatty acid oxidation itself by working on certain enzymes. So it's a very complex thing, but what I can tell you is that we can when we knock out fructose metabolism, the animals will live longer. And, even if they're not being fed fructose, they will live longer, showing that endogenous fructose production is important, is having a negative effect uh, uh, just in a normal mouse eating chow. Chow has about 50% carbohydrate. So they're eating a carbohydrate rich meal, but it has almost no sugar in it. But they will, it's been known for a long time that as you age, you get, kidney disease, and you can get, and mice do too. They'll get kidney disease. They'll become prone to hypertension, and we can block those effects just by blocking the endogenous fructose pathway. So we think that this is a really central mechanism. I would love to share with you our ideas of its role in Alzheimer's, because you're such a expert on this. You can, be a great conversation, and that we can discuss the merits and weaknesses of the hypothesis as well as the strengths. So I
Okay,
don't know.
let's do that. I'll just start throwing out one study a graduate student did in my lab, Alexis Stranahan, in
Yeah,
rats, and it was pretty simple. Have control animals and animals that have high fructose corn syrup in their drinking water, which they love.
great.
It's addictive to rats too, I think.
Yep,
And
that's right.
so she let them go for 8 months, right? And the
There you
ones on
go.
the high fructose got fat and insulin resistant. And then she looked at their learning and memory in these maze tests and found it was impaired. And she also quantified numbers of synapses, neurons in the brain region called the hippocampus, which is very important for learning and memory. And this chronic high fructose intake decreased the number of synapses. And that's correlated with the memory deficit. So that's just one example of an
It's a
experiment.
beautiful example, and actually, I have had friends who've done similar studies showing that when you give sugar, that it affects the ability of an animal to make it through a maze or a swimming maze or these different kinds of cognitive testing. And also there's national health surveys, what they call the NHANES survey, that kind of screened people and found that those who were drinking a lot of soft drinks tended to have more memory trouble. And there was the Framingham that showed some of the
Uh,
similar type studies. And there was even a study that showed that the more sugar you eat, the smaller your brain is by CT scan. So it's a scary one. And so here we are.
ah,
All of us love sugar, right? People love sugar. I mean, we go to birthday parties, cake is served, And some of us are strong and we go, we know that's bad for us, but we'll still have this craving for it.
You know,
And we'll end up, taking a small
you can,
piece and
you can kick the addiction, Rick. I haven't, I don't eat dessert at all for decades and decades, and it's interesting when you stop eating these, high sugar things, you start to taste like I just eat lettuce plain without any salad dressing and
yeah,
and vegetables plain, and I like it. Of course, I know from the research that these bitter tasting things are actually better for you than the
yeah.
sweet tasting things. So
Well,
I'm,
you're totally right. If you go on a keto diet after a while, or a low sugar diet, really low sugar, after a while, sugar tastes too sweet
yeah, yeah,
and almost and you do lose that craving. And one of the problems I believe with artificial sugars is that while they themselves do not cause these metabolic effects to the same extent, if any. And we've done a lot of studies, and I agree they can cause mild insulin resistance, but in general, the effect of an artificial sugar is way inferior. I mean, it does not cause obesity. Now, compared to a regular sugar, there's just no, we can't show that in any system. But when you're eating artificial sugar, you keep activating those sweet receptors, and so you never really lose your
yeah.
craving for sugar.
So you're
And
talking like saccharin and some,
yeah,
yeah,
yeah. So saccharin is the one where we find a little bit of insulin resistance with. We don't see that, for example, with some of the others. And we did do studies where we knocked out the sweet receptor. Actually, we knocked out all the taste receptors using kind of a P2XY, a
mm,
purinergic receptor
huh,
knockout.
yeah,
And when we did that, even though the animals can't taste sugar, they still like foods with, they still can identify foods with sugar, and they'll keep eating it, and they still get fat. And so what we found is that taste was good at increasing the uh, ability to find sugar and to eat more of it, but absence of taste did not prevent the metabolic effects. And the only way you could block it was to block the metabolism of fructose, as opposed to blocking the taste. Taste, you'll still get fat.
I had a question about, so there's GLP-1 analogs, GLP-1 peptides is all over the news, right? And
Yes.
anti-obesity effects. And so normally GLP-1, it's produced in cells in your gut, the epithelial cells in your gut.
Yeah.
When you eat a meal, and particularly sugar, carbohydrates, GLP-1's released into the blood. It increases insulin production, insulin sensitivity.
Right.
Uh, Couple questions then. Does fructose stimulate endogenous GLP-1 production and release? And then two, what about saccharin and GLP-1?
Yeah, I don't know about saccharin, but fructose does stimulate
Okay.
GLP-1 a little bit, as does glucose. There's a fructose mimetic. It's an epimer that looks like fructose that does not activate the pathway, and it's called allulose.
Yeah.
And it actually causes a bigger GLP-1 response, which, GLP-1, raising GLP-1 should be a good thing, right? Because the GLP-1 agonists are doing such great things. So you could say, why is bad sugar like fructose stimulating GLP-1? But it stimulates it a tiny bit. Whereas
Yeah.
when you take a GLP-1 agonist, you have blood levels 24/7 that are very high. And we actually think we understand a little bit why the GLP-1s work, and they are working in the brain.
Yeah.
And but I think that it's still unclear exactly how they do their benefit. But I have some ideas more than I have proof.
Well,
But
suppressing appetite is maybe a major one.
yes,
Yeah.
absolutely, and it does. And interesting, it works on areas of the brain where fructose it counters the effects of fructose.
Huh.
There's a paper on that. Well, it's getting back to the Alzheimer's story. It turns out that there is this big association of sugar intake with risk for Alzheimer's. There's a literature on that. And also on high glycemic foods, which remember get turned into endogenous fructose. And even there's some studies linking it with salt intake, and salt turns out to stimulate the enzyme that converts the glucose to fructose. So, like if you have a potato and it's not salted, it will not increase fructose as much production as when you salt it, because the salt, by raising the osmolality in the blood, will activate this enzyme, aldose reductase, that converts glucose to eventually to fructose.
That's
Um,
interesting. So salt's been so you mentioned that I think that high fructose intake or high glucose, which leads to endogenous fructose production, can cause hypertension. Do you think
yeah,
there's a role for fructose in the effect of high salt intake on hypertension?
yeah. Well, we had a paper in the proceedings of National Academy on this, actually, so your timing's good. What we did was we put animals on regular chow. These are mice, with or without salt, and we also had a group on salt that were mice that could not metabolize fructose. So we had the whole group of controls, and what we found was very interesting. When you eat salt, it raises the salt concentration in your blood, and you get thirsty. And initially, the response is to you eat more, but you also increase your expenditure. So weight doesn't go up initially, um, but you're eating more. And evolutionarily based, a lot of animals get water from food as well. And so when it probably is a response that they're not getting enough water, so they eat more food to try to provide that water. But over time, after several months, they start making a lot of fructose, and they also start becoming what we call leptin resistant, where they will eat more than they need, and it's a central leptin resistance. And then they'll actually become obese and diabetic and all those things, and their blood pressure goes up, and they get cardiac hypertrophy, which is often linked with high blood pressure. And when you knock out fructose metabolism and they're given exactly the same amount of salt, they get basically don't get hypertensive,
Huh. Yeah.
and their cardiac hypertrophy is much less. And so we began to realize that a salt may be working in part through fructose and in part through its osmolality. And so we did studies in people where we would give salt to people. And the way you can do that is you give them soup, and creamy soup. You can
Hmm.
change the salt concentration without them realizing how much it's changed.
Huh.
And we gave it with or without water, and we could look at things like blood pressure response and the activation of some of these fructose dependent pathways. And we were able to show in people that just by hydrating with water, we could block this acute development of hypertension, acute rises in cortisol, acute rises in ACTH, acute rises in vasopressin. And we found that there was a vasopressin 1B pathway where it was stimulating ACTH, cortisol, and glucagon. And so and that
Interesting.
seems to be important in all these pathways. Um, we also did studies where we knocked out vasopressin receptors, and we found that when you block this one receptor called the V1B receptor, we could block the ability of sugar to cause obesity and metabolic syndrome. So we think that there's a central vasopressin pathway, and it's through ACTH and cortisol that's involved in obesity and metabolic disease. Anyway, but getting back, what we found in the Alzheimer's case was that, and a lot of this was from the literature. It wasn't all from our group, but what was known is that in Alzheimer's disease, you get amyloid plaque and tau protein aggregation and neuronal loss and cortical atrophy, and everyone's been focused on the amyloid,
Yeah.
rightfully so, because this really characterizes this disease. But blocking amyloid seems to be coming in a little bit late because it's not working as effectively
Yeah.
as they were hoping. So then what people are saying is, okay, well, let's look a little bit earlier in Alzheimer's to see if what precedes the amyloid, maybe we need to hit it earlier. And they found that there were actually four major characteristics, and you'll probably correct me because you're brilliant and know this disease better than I do, but the four key characteristics that we saw or that I found in the literature were mitochondrial dysfunction. The
Yeah.
mitochondria aren't working as well. And associated with that, they're not producing as much ATP as we would like.
Yeah.
There's insulin resistance occurring in the brain,
Yeah.
and there's neuroinflammation. And so neuroinflammation, mitochondrial dysfunction, insulin resistance, and low ATP.
And oxidative damage
Yes,
accumulation.
we can throw in the oxidative damage, which is often mitochondrial.
Yeah.
And guess what? When you give fruit, all my work in the systemic systems showed that fructose was causing mitochondrial dysfunction, mitochondrial oxidative stress, and regular oxidative stress, low ATP, insulin resistance. And it was like the driver in all of our models. It's like the driver of insulin resistance and in Western diet, sugar diets, glucose diets. And so we said, oh my God, it's the same bioimprint. And then we started putting it together that it had to be endogenous fructose in the brain that was driving it. And basically, in the brain, there's a cell, the microglial cell, which is kind of like the monocyte, the protector cell, and it carries this enzyme, and it gets inflamed when the microglia is fed fructose. And there's now three groups that have looked at this, including one with us. And then what happens is that it leads to this state. And if you give fructose to a mouse, Gomez Pinilla, who you probably know, Pinilla,
Yeah.
showed beautifully that you get mitochondrial dysfunction, insulin resistance, all these changes, and it hits the areas like the hippocampus and those kinds of areas. And then if you keep giving sugar, this group in Egypt found that if you gave it for like 18 months, the brains of rats starts developing amyloid plaques and tau protein. So it's kind of like, whoa. And then if you look in the brains of patients with Alzheimer's, they have high fructose levels, like 5 to 6 fold higher. They have high sorbitol, showing endogenous fructose. They have elevations of these enzymes like AMP deaminase. I mean, it's there. And then recently, a group showed that if you knocked out fructokinase in the brain, you can block cognitive dysfunction in a diabetic animal. And so I think, and then the cinch of it was we looked at the evolutionary pathways, and we realized that fructose stimulates foraging. And actually, you've done some work where you show that uric acid stimulates impulsivity and locomotor activity and be able to exercise.
Yeah.
This is like a locomotor activity. And when you give fructose to people, and you do like brain MRI and these kinds of tests, you'll see that it lights up the occipital cortex for visual cues for like delicious foods and so forth, so you'll spot them.
Yeah.
And at the same time, it suppresses cortical blood flow, which is good because it reduces self-control, which you need if you're going to be foraging. And it stimulates, it inhibits the hippocampus, so it blocks recent memory. So you don't quite remember that lion that's out there, because what it stimulates is foraging. And so this is a foraging response. You give fructose, you get a foraging response. It's supposed to be beneficial. It's going to help you find food. So you become impulsive, less self-control, a little bit of locomotor activity. And guess what? Those areas that are inhibited to allow this foraging behavior, those are the same areas where Alzheimer's develops. And the areas that
Interesting.
get increased blood flow, like the occiput, they're typically spared in Alzheimer's. And
Yeah.
when you see a patient with Alzheimer's, they develop this thing called a wandering complex or wandering syndrome, where like maybe half of them will like wander off and you don't know why they're doing it and they're kind of wandering off. And that, I believe, is a carryover with the foraging response.
You
So,
know,
yeah,
that's
go
very
ahead.
interesting from a brain evolution standpoint too, because our our non-human primate ancestors, the earlier ones, pretty much lived in the canopies in the forest and they foraged on fruit.
Yes.
So you can imagine there's a tree with cluster of fruit there, and some of them are ripe, some are not. And so the monkey eats all of the ripe ones, which have a lot of fructose and glucose, and then when when they run out, when they're done eating that, then the fructose, what you're saying, the fructose does things in the brain that cause them to leave where they are and try to find another place with
Well, yeah,
more.
let me expand on that. So first off, primates love fruit, and they have a craving for fruit that's above and beyond what people, what many other animals have, and it's been written about. And the orangutan, for example, will will eat a lot of fruit in the wet season, and then it will gain fat that will help it survive the dry season. And the
Yeah.
lemurs will eat a lot of fruit to increase their fat stores, and then they'll do a thing called estivation. It's sort of like hibernation, where they burn off the fat mainly to produce water, actually, um, and calories. So there's all this link of fructose, but interestingly, remember that fructose generates uric acid, and some of these effects of foraging appear to be linked a little bit with uric acid. And there was a mutation in uric acid metabolism that occurred around 12 million years ago. And it occurred during a time of starvation, uh, when there was a global cooling. And I got very interested in this, and I contacted a number of anthropologists who were studying prehistoric primates and the evolution of humans. And I finally I talked to Pilbeam, but I also reached out to Peter Andrews, who was at the Museum of Natural History in London. He was like the world expert on this period of time, and he had published in top journals showing that what happened was, when global cooling occurred, uh, the oceans receded and polar ice caps
Huh.
formed. And Africa, which had been basically separated from the other continents, developed a land bridge. And around 17 million years ago, that land bridge led to an exodus of animals from Africa into Eurasia. And they included anteaters and giraffes and also primates. And there was these early primates like proconsul. They were basically the earliest apes, and they made it into Europe. And then global cooling continued. And what happened is, as global cooling continued, the rainforests and the forest that these animals were living in started to get more and more sparse.
Mm,
And the animals actually had to come out of the trees
yeah.
and forage for foods and look for other types of foods like tubers that they never ate. And this was a time of very rapid evolutionary change because there was this dire environment. So the teeth became stronger to eat more harder foods. The animals started becoming upright. They started becoming bipedal, where they would walk just on their legs, and it was a very active time in evolution for our ancestors. And what Peter Andrews showed was based on the evolutionary fossil record that humans are descended from one of the European apes, not from the African apes of the time. And he actually identified some of the species. He did a lot of work on it. And he showed that those European apes went back to Africa in this
Huh.
as things got colder and more horrific. All the apes in Europe eventually died, but some of them made it back to Africa, and some bearing the same mutations made it to Southeast Asia to become the orangutan. So all the great apes and humans have a common ancestor that he was able to link to Europe during this starvation. So I flew over to London to meet with him. Fantastic guy. We spent a lot of time talking about the mutation that occurred in uric acid metabolism 12 million years ago and how that had to be carried by the common ancestor. So it had to have occurred in Europe. And so we ended up writing about this and called it, it was like a thrifty gene, because here's a mutation that helped them forage, that helped them convert fructose to fat, and by knocking out this gene, it led to a higher uric acid level in response to fructose. So when we
It,
published this, the people said, well, can you give us proof? Can you show us that that actually is the case? So what we did is we did two experiments to prove it. The first one was we took rats, and normally to make a rat fat, you have to give it a lot of fructose. And some people say, hey, it's not fair because they're eating much more fructose than the human does. But when we inhibited this gene in uric acid metabolism, so they had a higher uric acid, then even low doses of fructose could cause them to develop metabolic syndrome. So that was the first one. And then we actually, with Eric Gosher and Steve Benner, we resurrected the extinct gene and did studies in cell culture and so forth to show that originally the animals were much less sensitive to fructose, but after this mutation, they were much more sensitive to the obesity effects of fructose. So the truth is that today we all carry this mutation. We not only do we love sugar, but we can convert it to fat much easier than most animals. And we're we're eating a huge amount of sugar. We're making a lot of fructose, and we are super sensitive to fructose. So it's like the perfect storm. All driven by evolution.
so humans have much higher uric acid levels than some, lower primates, before
Yeah.
this mutation in the uricase enzyme. Now, but there's some interesting, I did a lot of work on aging. I was at the National Institute on Aging for
Yeah,
20
you were
years
the head of it.
of the neuroscience.
Yeah.
Yeah.
Okay.
And so if you look at there's a correlation between plasma uric acid levels and longevity, lifespan among different species. Species with high uric acid levels tend to live longer. Humans live longer than non-human primates. Bats and birds, particularly like corvids and parrots, they have high uric acid levels. They live much longer and that, even though they have a high metabolic rate. And then also there's real good evidence that uric acid is a free radical scavenger.
Yeah.
So then this raised the question, the work with fructose says that uh, high fructose, either eating or coming from endogenous, increase uric acid levels and does bad things. And
Yeah.
if you if you put the uricase back in a mouse or rat or whatever cells, then the effects of fructose is lessened. So then there seems to be good and bad
Yeah,
aspects of
yeah,
uric
and you've
acid.
done some nice papers on this. Yeah, let me try to address this because it's a very common um, area where there's a lot of debate. So the first thing is that when we actually went to look at uric acid levels in primates, I went to the San Diego Zoo, and we also got samples from um, of hunter-gatherer groups that were still living in hunter-gatherer situations. And what we discovered is that if you lost the uricase gene, which is what happened to all of our ancestors and the great primates, the great apes, that only took the serum uric acid from 1 to 2 milligrams per deciliter to 3. And in fact, like the Yanomami Indian had a uric acid of 3. And most of the primates, like chimpanzees and gorillas, they have a uric acid of 3. Our average uric acid in our country is like 6, and there are people with levels of 12, 14. I mean, it can go all the way up. So what happened was I then also looked historically in the literature at uric acid in Americans. And like in 1920, the average uric acid was 3.5. 4 was
Yeah.
considered high. And then by every decade, normal is upgraded to a higher level. And it just keeps going up. And in fact, papers are coming out now. Gout's occurring in adolescence, and the serum uric acid is shifting. Now, when Ames, who's this wonderful person who studied oxidative metabolism and oxidants and antioxidants,
Bruce Ames. Yep.
Bruce Ames, great guy, wrote this beautiful paper showing that uric acid could be an antioxidant. And he noted that, just as you did, that if you look at many species, animals that tend to have higher uric acid tend to live longer. And so he published his paper, which has been cited, thousands of times, and basically says, uric acid it was probably good. And this mutation may have replaced the vitamin C mutation
Huh.
that we lost, where we lost vitamin C, and now we have high uric acid, and it's an antioxidant, and it should promote aging, and people should live longer. But in fact, as you know, high uric acid levels in humans are associated with earlier death. And in fact, a beautiful centenarian study was published, I think from the Aging Institute, where they showed that when those people who made it to 100, they went back to the age of 65 with
Ah.
the big cohort.
It's a Baltimore longitudinal study.
That's it.
Yeah, yeah.
And the one of the most prominent uh, things that came out was that if you had a low uric acid at age 65, you had a dramatically increased risk for living to 100. Not the reverse. It's the opposite of this aging Ames. Ames.
I don't know. I've never had my uric acid levels measured, I don't think.
Oh, you should. But I bet you yours is pretty good. You know, I
Well,
bet you yours is.
what about acutely? We've done all this work on intermittent fasting and
Yes.
caloric restriction. Can you should be able to lower your uric acid through diet, eating pattern, and what you
Yeah.
eat. Yeah.
Yeah. So, well, anyway, let me just finish with one thing. If you mix uric acid with oxidants, it will block peroxynitrite in particular. It is very, very effective at that. So in an in vitro system like what you've done, if you create oxidative stress and you add uric acid, it will block oxidative stress so you can get some benefits. But when it gets into the cell, it actually stimulates low-grade intracellular oxidative stress. So it's not a 100% win situation. And we found that when uric acid reacts with peroxynitrite, it actually generates some radicals in the process too. So it's not all benign, and but acutely in an extracellular environment, uric acid is an antioxidant. Now, getting to your other thing, well, there is this very conflicting area of science that is, which is like, you go on a keto diet, you're getting this huge benefit, or intermittent fasting, as you are the world expert on. But if you go any kind of fasting or a keto diet, it's not uncommon for uric acid levels just to rise despite getting all these other benefits. In the keto diet, we actually know that when you start making beta hydroxybutyrate, that it blocks a lot of the effects of uric acid,
Mm.
that anti-inflammatory effect. And we did studies and published that beta hydroxybutyrate blocks uric acid induced inflammation in vitro. And so and remember that one of the, but well, I haven't really talked about this, but just like salt can activate the mechanism to convert glucose to fructose, uric acid also activates that pathway.
Mm.
But if you're on a keto diet, you don't have enough glucose to convert to fructose. So that amplifying effect of uric acid to kind of make more fructose is blocked because you're on a keto diet. And if you're intermittently fasting, you're also reducing your carb exposure during that time at least. So anyway, it is true that uric acid can go up acutely with keto diets and with fasting, and then it tends to come down after a while. But there are a few people where it will stay persistently high, and it can cause gout in people on keto diets, and it's not all benign, but it
So,
is
so the gout,
present.
Rick, the gout. So my understanding is that with gout, the uric acid gets high enough levels that it starts to precipitate out and form these crystals and that accumulate in the joint, and then you get inflammation locally. What's what's the concentration that would be sufficient to cause it to form crystals?
So there is a general relationship of serum uric acid with risk for gout, and it starts to go up when the serum uric acid is 6 or 7.
Hmm.
And then it gets very high when it gets up around 9 or 10. But it isn't a perfect relationship, and there are a lot of people who have a high uric acid who never get gout. And so there seem to be other factors involved in the crystallization of gout. But what we do know is that, like, classically, if you eat a food that's rich in purines or sugar, or alcohol, these are the three foods that cause that rapid rise in uric acid. And so if you drink like beer, which really is high in purines, certain seafoods that have a lot of RNA and DNA, they create these, they can raise your uric acid acutely. So you eat lobster and beer and then have a big sugar dessert and you go to bed and 2 hours later you get an attack of gout. That probably is related to the rise in uric acid post meal, where it seems to trigger some of this crystallization. But there are other factors too, but that is the classic one. And one of the scary things that has happened in the last 5 years has been the recognition that although the joints are the favored place for crystallization, and also it can sometimes form under the skin, what we call tophi, but it can also precipitate in the kidney and in other organs. And one in particular that has received a lot of attention is it likes to deposit in atherosclerotic plaque.
Mm.
So like in the coronary arteries that are where it's
Mm.
diseased, and the aorta. And now it's being, the uric acid deposits in the atherosclerotic plaque are being correlated with increased risk for cardiovascular events. What has not been shown is whether or not removing the crystals from the plaque would help, but we do know that the plaque, the crystals stimulate a thing called inflammasomes, which are activated in atherosclerotic plaque and which are thought to cause disease. So the odds are likely that if you do have uric crystals in your aortic, your aorta, or your coronary arteries, that it's probably not doing any good.
Yeah.
And one of the things that I recommend to everyone is if you don't, I love intermittent fasting, by the way. But keto diets, intermittent fasting, uh, if not that, just try to eat a healthy diet with reducing the amount of carbs,
Yeah.
salt, and sugar. Sugar is a carb. I realize that, but cut back on the high glycemic carbs, on the salt, on the sugar. Try not to eat the super rich foods that can raise uric acid. Um, Seafood is the classic. Certain gravies can be bad. Brewer's yeast, which they put in the beer.
Let me, one thing we did, we had a senior uh, medicinal chemist in the group.
Okay.
And we had this idea and did it that since uric acid is quite insoluble, and the reason we did this, we'd previously done some experiments where we infuse acutely uric acid intravenously into rats and then do a stroke model and found
Yeah.
some neuroprotection.
Right.
But it was hard to get enough uric acid. So what we did is we added methyl groups to the uric acid molecule to make
Uh-huh.
it more soluble. So we had like three different analogs, and so their solubility was increased like 100-fold or more.
Wow.
And we found that several were neuroprotective in some of our models. But the one thing that's interesting is one of them, that's 1,7-dimethyl uric acid. It's a natural metabolite of caffeine.
Yes. Yes. And it's also inhibited the formation of um, it's inhibited the caffeine metabolite is inhibited by allopurinol.
Oh, interesting.
And there are these nice studies. So again, you're looking at the acute effects,
Right.
which are an antioxidant. And when you give it acutely during a stroke, it may actually decrease the severity of the stroke, as you've shown, and Chamorro and other people have shown in humans. But it comes with a cost because the chronic effects of high uric acid are clearly bad.
Yeah.
And people with chronic hyperuricemia, there's a lot of studies suggest they may have an increased risk for stroke. So even though um, uh, the acute effects of uric acid could be good, we have to be careful.
What
But yeah,
about that? What about the epidemiological studies indicating that people with higher uric acid levels are at reduced risk women for multiple sclerosis? I've seen multiple.
yes, there are studies showing that I think that hyperuricemia uh, may have may block blood-brain barrier, may make the blood-brain barrier tighter initially because it sort of inhibits nitric oxide. Um, but chronically, it may not be so good.
Oh, maybe it's because it's an inflammatory, it's an autoimmune disorder. Maybe the high uric acid is suppressing or somehow altering
Yeah,
the immune reactivity with
yeah,
the,
yeah.
I don't know.
But I do know like the studies giving inosine and things like that were not beneficial.
Ah,
And they, I think that there is this, nice relationship where higher uric acid is associated with a lower risk of Parkinson's, for example, as well. But I think the clinical trials suggest that it's not modifying the uric acid or by raising it, you're not helping those diseases.
okay, yeah, yeah. Let's finish up. So we haven't really talked about, it was kind of
Yeah,
implicit in everything we've talked about that the time course of increase in fructose intake, when did high fructose corn syrup infuse
yeah,
the food and drink market, and the temporal relationship between the big rises in obesity and related diseases
Yes.
and so on.
Yes.
Yeah.
Yes. So if you look, there has been a general relationship of sugar intake with obesity, diabetes, and all these diseases just from sugar intake. And I've done graphs showing that, we really start seeing the rise of obesity and diabetes beginning in the 1890s and around 1900. And it was
Oh,
rising slowly with dips during the world wars because there was less sugar ingested. And everything was kind of going at a slow rate. And then around 1970 or so, uh, there was this discovery of a way to make um, fructose from corn.
yeah.
And basically, it's an enzymatic pathway. And the big breakthrough was that there were all these enzyme reactions, but they found that if they fixed the enzyme to like a column or to something where the enzymes wouldn't be, before they would mix them and then you'd lose the enzyme, but
Yeah.
they fixed the enzyme so you could pour the material over it and the enzyme stayed stuck on the column or stayed on uh, the absorbent or whatever, so that you could make a lot of high fructose corn or of fructose without losing the enzyme. So it became financially feasible to do it and to do it in an industrial levels. And so in the late 70s, there was the introduction of high fructose corn syrup. And high fructose corn syrup is kind of like table sugar. Table sugar is one glucose, one fructose bound to each other. And when they were making the high fructose corn syrup, they could, because they were making fructose from glucose, they could mix the two to identify the ideal concentration that people liked. And they did all these tasting tests and they found that the 50-50 was great. But if they made it 55 fructose and 45 glucose, it was even better. So they realized it was even more attractive than sugar in many ways. And so they started making soft drinks, and George Bray, who is this, and Barry Popkin, these guys who were studying the epidemiology of obesity, saw that there's this huge inflection that correlated with the introduction of high fructose corn syrup. And in terms of how that worked, when the high fructose corn syrup was introduced, the theory was that it was going to replace sugar, but it didn't replace sugar. It added to sugar. So sugar intake did drop by maybe 30%, but the increase in high fructose corn syrup led to a net effect that the sugar intake was the intake of both sugars was went up dramatically. And so suddenly you see this big influx, this big rise in obesity and diabetes and chronic kidney disease and all these things that are linked with with sugar. So that's what happened.
I had a fellow in the lab, and her and I wrote an article for Trends in Neurosciences on potential role for high fructose intake in risk for autism. In other words,
Oh yeah.
women during pregnancy, there's good evidence that women with obesity and insulin resistance during pregnancy, there's increased chance their child will be diagnosed with autism. And that, if you map it, like you're talking about mapping it onto the fructose, the increase in incidence of autism lags just a little bit behind the increase in fructose intake, which would make sense. And there's some mechanisms to explain that having to do. One thing, with intermittent fasting, exercise, we've done a lot of work in this idea that health can be optimized by challenge recovery cycles. So
Yeah.
fasting recovery,
Our immune system.
exercise recovery. But if you stay in a constant positive energy balance, and particularly maybe with fructose, then the cells, they're continuously in this like growth mode, and they don't upregulate their endogenous, for example, autophagy or protective
Right.
pathways. Yeah.
Yeah, no, that might be important not just in general biology, but in conditions like hibernation and things like that. I do understand this, and yes, I do think this association with autism is uh, very interesting to me. Sugar has been linked also with ADHD and with bipolar disease in particular, but autism is such a major issue now. Yeah, I think everybody can benefit by trying to do
Maybe
intermittent fasting
you
episodes.
need to contact somehow RFK Jr. and because it's maybe the one sensible thing he seems to be doing is
Yeah,
trying to push, eliminating or not eliminating or at least reducing ultra processed foods and
yeah, that would be a great benefit, a great thing that he could do that would really help the health of our nation. So um, I would love him to uh, be pro on vaccines, but at least childhood vaccines, but where I think they're really important. But I do like the fact that he recognizes that there's a lot of um, ultra processed foods are doing no good and are making things worse.
yeah,
And eating healthy and reducing sugar would be a great thing. So I support him on that.
yeah. Uh, okay, let's see. Man, we covered a lot.
Yeah,
So, take home message, quit eating, quit ingesting fructose.
yeah.
Yeah.
Well, I do want to make a correction to that.
Okay.
Natural fruits contain a lot of good things in them.
Yeah.
Vitamin C, um, flavonols, fiber, potassium. There are even these things like the flavanol called epicatechin that actually can counter some of the effects of fructose. So it's my belief that natural fruits are healthy. And in fact, we did a clinical trial where we put people on a low fructose diet with or without fructose supplements and fructose supplements. I mean, with fruit supplements and natural fruits supplements. Actually, they tolerated the diet a lot better and overall probably did better. And so I believe that natural fruits are
Yeah.
good. I think if you eat a huge amount at one setting, that's not good. Fruit juices, it's concentrated,
Ah.
it's probably not good either. But natural fruits are something you should eat if you really want to eat something sweet. That would be a great choice.
A lot of the good healthy chemicals in those fruits are actually in the skin.
Yeah, exactly. Quercetin, for example.
Yeah.
Yeah. That's a really good point. I like that.
Yeah.
And I'm a big fan of vitamin C, and I recommend taking 500 milligrams a day. I don't think you need to take the huge doses because of especially if you have a history of kidney stones because it can generate some oxalate. But yeah, I
I
love it.
don't think it hurt Linus Pauling much. He lived to 93 and
Exactly.
yeah, so
Yes.
and he was taking what 10 grams a day or some.
Yeah. A lot of people tolerate high doses pretty well, for sure.
Actually, it would be since it's vitamin C is like I could say disappears pretty quickly after you take it, so it might be better to take like space it out a little.
Yeah, it's a good idea.
Yeah. Oh,
Yeah, you're filled with great information. I love it, Mark.
well, likewise, I learned a lot, and I'm yeah, the uric acid story is really fascinating to
Yeah,
me,
it
and
is.
yeah, yeah. Okay,
Thank you
well,
so much.
yeah, thank you, Rick.
Yep. I'll send you a paper I wrote on uric acid as in thrifty genes that kind of describes a lot of the literature. And
Okay,
you're welcome to share it
great.
with
And
others.
I don't know if you saw, but I'll send you this paper on the autism relationship
I
with.
would love to see that.
Okay.
Okay, thank you.
Bye. Have a
It's been a
good time
pleasure.
in Colorado. It's a great state. Me and my wife were there for 3 years while I did a postdoc at Colorado State up in,
Oh, yeah, beautiful.
yeah, yeah,
All right. Well, yeah, it is a beautiful state. Take care.
yeah. You too. Bye.